As we finish up our discussion on carbohydrates, I would like to touch upon both glycosylation and glycoproteins. In our previous videos about carbohydrates, I mentioned that carbohydrates function as energy molecules and also provide structure to cells as well. Another function that carbohydrates have is that they are able to increase the function of proteins. Proteins can be modified in such a way through the process of protein glycosylation, which occurs when a carbohydrate is covalently bonded to a protein. And as a result of this carbohydrate addition to the protein, this type of molecule is known as a glycoprotein. One important thing to note is that there is another molecule that consists of a protein and carbohydrates, and this molecule is known as a proteoglycan. But it's important to understand that proteoglycans and glycoproteins are not the same type of molecule. The difference between the two is that in a proteoglycan, the molecule consists of a higher percentage by mass of sugar than does the glycoprotein. Now going back to glycoproteins, they are molecules that can be found in the cell membranes as well as the extracellular matrix. Now I want to discuss the process of protein glycosylation, which is the addition of a carbohydrate to a protein, as I mentioned earlier. One type of carbohydrate molecule that may be attached to a protein is known as an oligosaccharide, which is basically a carbohydrate molecule that consists of a relatively small number of monosaccharides. And an oligosaccharide can be attached to proteins through specific amino acids, namely serine, threonine, and asparagine. When an oligosaccharide attaches to either of the three amino acids, there are only two types of bonds that can form. The first type of bond is known as an O-glycosidic bond, which occurs between the oligosaccharide and the oxygen atom found on the side chain of either serine or threonine. The second type of bond is known as an N-glycosidic bond, which occurs between the oligosaccharide and the nitrogen atom found on the side chain of asparagine. So I want to show you visually what an O-glycosidic bond and what an N-glycosidic bond looks like. So in the case of an O-glycosidic bond, the oxygen atom of the side chain in either the serine or threonine amino acid is covalently bonded to the first carbon of the carbohydrate, which is labeled right there. In the case of an N-glycosidic bond, the nitrogen atom of the side chain in the asparagine amino acid is covalently bonded to the first carbon of the carbohydrate labeled right here. Now, the way that the cell knows which amino acid needs to be glycosylated depends on the amino acid sequence around either the serine, threonine, or asparagine amino acids. And the way that the cell knows which amino acid needs to be glycosylated also depends on the structure of the protein that's being glycosylated. I want to zoom out a little bit and discuss which organelles are involved in the process of protein glycosylation. Now, the organelles that are involved in this process are both the rough endoplasmic reticulum and the Golgi apparatus. So here we have the rough endoplasmic reticulum, and then here we have the Golgi apparatus. And then this is the cell membrane. So I discussed how there are two types of bonds that can form between the oligosaccharide and the amino acid, which are both the O-glycosidic bond and the N-glycosidic bond. Now the N-glycosidic bond begins in the rough endoplasmic reticulum and then later moves to the Golgi apparatus where it is completed. And the O-glycosidic bond is formed solely in the Golgi apparatus. Now at the site of the endoplasmic reticulum, which is covered in ribosomes, protein synthesis takes place and the amino acid strand moves into the inside or lumen of the rough endoplasmic reticulum. So I'll draw one ribosome for simplicity's sake. And there's a amino acid. So this is the lumen, or the inside of the endoplasmic reticulum. And so inside the endoplasmic reticulum, there are enzymes that form the N-glycosidic bonds between the oligosaccharide and the asparagine amino acid. And once the N-glycosidic bond is formed, the molecule leaves the endoplasmic reticulum. It leaves the endoplasmic reticulum and heads towards the cis phase of the Golgi apparatus via vesicle. So it will then travel via vesicle to the cis side of the Golgi apparatus. And as the molecule moves to the Golgi apparatus, the N-glycosidic bond is modified even more until it is completed, and the O-glycosidic bonds are formed here as well. Now once the glycoproteins reach the trans phase of the Golgi apparatus, which is this side, trans side, the glycoprotein can be transported to various parts of the cell. Now the location in which the glycoproteins are sent to depends on the structure of the molecule, and so the glycoproteins can be secreted from the cell through the process of exocytosis. They can also be placed into an organelle called the lysosome, which are organelles that contain digestive enzymes, or they can be sent to the cell membrane to be stored there.